Hi, so my name is John Lees, and I'm the group leader of the Pathogen Informatics and Modelling Group at EBI. And today I'm going to give you an introduction to computational and statistical modelling, also sometimes called mathematical modelling. I'll explain some of the different terminology in the video, but they're all kind of the same thing here. Today, unfortunately, only time for a brief overview of what's of course quite a large field, so we've got I think about 45 minutes. I'll try and overview a few different kind of key parts so that hopefully you get an idea of at least what this is all about, and then also if you think it might be something that you could employ in your own research, at least give you a start of the kind of things you might look at to use in your own work. Also, just like to say I'm sorry for not being there live and virtually. I'm away this week. But if you do want to talk about any of the material here today, I'm happy to set up a Zoom call or just answer via email, or if there's a group that wants to discuss, I'm happy to do that. So please do reach out to me by email after the session. So I just thought I'd give you a quick introduction of what our group at EBI works on. So we work on infectious diseases broadly, but I suppose mostly looking at bacterial pathogens, although not one species in particular. And we kind of had two main areas of the group. So the first one is using and developing informatic methods to deal with large amounts of usually genome data, but other kind of omics data, and processing large public databases or sequence data sets, either looking at relationships between them or functional information in them. There's a couple of examples there. And then the other part of the group, which is more what I'll talk about today, is to create models, usually evolutionary models, using that data as input. So there's a couple of examples listed there. We'll kind of go through some of these briefly, but again, if you're interested, have a look at our group website, which is linked at the bottom there, and a couple of the papers linked for some sort of introductory examples of how you can link these two things together. So I guess these are kind of the rough goals for today. So the first is just an introduction of why you would want to use computational models. I think the world of modelling can be somewhat mysterious. I certainly found this until relatively recently. I wasn't sure exactly what research modelling looked like and how you would do that. So hopefully some of this, if you feel the same, I mean, you may not, but if you feel the same, hopefully some of this can kind of demystify what it is and how you might get into it. And hopefully I'll give you some confidence to use this in your own research or explore it further. It's perhaps a less well written down field, I would say, than bioinformatics. Everything is a bit more bespoke. It's possible to do more yourself, for better or worse. And so you do need some level of background understanding to know how to get started with it. But ultimately, I think it's no less approachable than bioinformatics itself, even though perhaps using a single package of software to do your analysis for you is a bit less feasible with modelling due to its very sort of bespoke nature. So that's the overall goal, and the sort of three main areas of trying at least touch on is how you go about designing models, how you fit the models you've designed to your data, and then also more throughout than in a special section, give you some ideas of packages you can use to help you with modelling rather than having to do everything yourself from scratch. I also think that you'll get these slides afterwards, so I've also got a list of resources at the end for further reading. So firstly, just kind of a very broad question. Here's a perhaps bad answer to a question of what is a model. So any simplification or abstraction of a system that makes it easier to study. So that is, of course, very vague and generic. And basically everything in science, every hypothesis, any description is a model. It's not exactly reality. There are probably some examples of things which aren't models in science, which is interesting to think about, but I'm not going to touch on that today. maybe to try and make this a bit more concrete, here's a few examples of things that are models, or different types of models. So physical models. So a physical model would be the chemical structure of DNA. That's a model of what DNA is. It's not exactly what DNA is. Actually, making it up from polymod, the little plastic balls, is also a model, because it's not actually not the DNA, but it makes it kind of easier to see and study. A very common use in biology of model is animal models. So a mouse is, of course, a very popular animal model. And here your model is that the animal is a human. So we do experiments on mice, we find things to be true, and we say this happens in mammals, but particularly we're interested in humans. And obviously mice have enough of their biology the same as humans for that to be a reasonable model, but of course they're not the same as a human. Statistical models are something I'm sure everyone's done before. So here's an example of a chi-squared test. So here you assume that your statistics follow certain distributions. The data points you've collected obey certain rules. So for example, that they're independent and identically distributed. And if you make these assumptions, you can model your data as having certain distributions, but of course they don't really have those distributions, but you can solve them quickly, and you can get p-values, for example, by making these assumptions. And then really what we're going to be talking about today is a sort of very overall class of models called abstract models, but basically things you can write down as equations, and really kind of a subset of these mathematical models or computational models. So they've historically been called mathematical models just because they have equations somewhere in there, but sort of the modern use and the use in biology we're going to talk about more are computational models, so where you use a computer to solve those equations or simulate them. So the example I put up there is actually Newton's law of cooling, which is an equation for temperature of systems, but we'll go on with some more biological examples. Sometimes you'll hear people talk about, I guess modelers mostly say this, there's kind of two types of scientists. There's scientists who write down their models, and scientists who don't. So that's kind of saying that everything in science implicitly uses a model, and you can either explicitly write it down, or you've just assumed it and you have the model somewhere in your head or in kind of text as a hypothesis. And I'd say that's a slightly glib response to people saying that modelling isn't like a real experiment and real data. I think that opinion is kind of slightly fading. I think modelling does get more acknowledgement than it used to, and also that anytime you do experiments in the lab, you are of course using some sort of model as well. So modelling, very general and in science, but we're going to talk about computational and statistical modelling today. So kind of just to briefly go over this, I mean, models are basically a way of, mathematical models synthesizing some evidence, data, observation, sequencing data with your biological knowledge, a mechanism which might be from prior research, it might be a hypothesis that you have, but it kind of forces you to be explicit about how you relate these things together. So you create a model which synthesizes both of these things together. Usually you go round in cycles, so you start with the first model, you see whether it explains the data well. If not, you change your model slightly, you update some of your knowledge until it does fit the data well. And then using this, you can do some pretty useful things. So you can, if you've got parameters in your model, for example, like the binding rate of two substrates, you could try and infer that rate from your data. You can do what-if scenarios, which are normally called counterfactuals in modelling. So, for example, what if I had vaccinated people with this formulation rather than this one? So they're called counterfactuals because they're things that didn't actually happen. So you're saying, if this had happened in the past, what would have happened? And then the final thing, which my picture is covering, is forecasting or doing predictions, so going forwards in time and saying what will happen to this system in future, usually assuming that the system doesn't fundamentally change. So here's a quick example of sort of familiar but complicated example. So this is from COVID-19 modelling in the UK. And here you've got all these different data streams. So numbers of cases, numbers of people going in hospital, numbers of seropositivity, numbers of PCR tests. All of these go into this model in the centre here. And in here we have our biological knowledge, basically our knowledge of immunity to COVID, and also about how people progress through hospitals. But that's kind of a complex bit that integrates all of this data together. So all of the different data can come in here, and then you can get these useful predictions out. So to show an example of that, here's a prediction of R, which is the basic reproduction number over time, RT. And this line 1 is important because if R is below 1, then the epidemic, the number of cases, is decreasing. If it's above 1, then it's increasing. And we knew with COVID that if the epidemic was increasing, the hospitals would be overwhelmed, and that was an untenable situation. So you can see here in the UK, there was this period towards the end of the summer where the epidemic was no longer under control. Now this is, if you look at the raw data here, you would see the cases which are very noisy due to reporting delays. You see fewer cases being reported at the weekend, for example. They'll be from PCR tests, which are partial and very noisy. So actually saying what the current reproduction number is would be impossible without a model, but kind of synthesizing all of that knowledge together makes it possible to get these very practical and qualitative insights out. It is probably the ability to do this that's made this kind of modelling so popular with policymakers. Just to give another kind of toy example that I'm quite fond of, which is kind of emerging and hasn't been done much in the past but is now becoming possible, is trying to predict the impact of vaccination. So consider a pathogen population like this. This is kind of, it's a simplified example, but this is not dissimilar to something like Haemophilus influenzae. So you have 3 different variants of the pathogen circulating in the population at some start time where you start looking at how many cases of infection you have, and then you're able to develop an effective vaccine against the most common one of these. So you know the vaccine is effective, so at this time I'm going to vaccinate against the orange cases, and those cases are going to go down. But kind of by ecology and also experience, we know that what's going to happen is that the other variants are going to fill the gap in the population left by this kind of currently dominant strain, because they're no longer going to have that to compete with. So we know these other 2 variants are going to go up in frequency. But if one of them is benign, doesn't cause disease, and the other is bad, I mean, potentially even worse than the orange variant, maybe it causes more severe disease, maybe it's resistant to other forms of treatment, it's kind of really important to distinguish between these 2 scenarios. So which one of these variants is going to grow? Will it be the benign one, in which case, great, you know, that's a very successful vaccine. If it's the treatment resistant one, for a period this will be a successful vaccine, but then in future the population, the amount of disease burden will just be restored to what it was before, or it may become even worse. And then this becomes particularly important for cases like this, where you have a really complicated population with many variants, hundreds, sometimes even thousands. Streptococcus pneumoniae, for example, is like that. Again, you can have this vaccine against one or even more of the current variants, but being able to predict which of the other variants will fill this gap in the population is important to design future vaccines and also inform rollout. And the way of doing this is to incorporate some knowledge about immunity, also observations from genome sequencing into these models show that you can predict accurately forwards in time, and then run these models. So I think this is the final example. So this is a kind of classic example from biochemistry. So this is the MAPK cascade. And here, this is a paper from 1996, which I think is the first one to describe this. These authors were able to write down, you can see on the left, quite a long system of equations to describe the different steps in this cascade. These equations in biochemistry are equations on the rate equations happen, so these become differential equations. And so this is kind of the model of the system on the left, and then the authors show that they can predict different steady state, and also actually dynamic trajectories of different parts of the substrates in this reaction. So you may have come across biomodels, which is the database of these EBI. This is kind of, I think, originally a database of these kind of biochemistry models, but it's been expanded. There's models of tissues, of ecosystems, including these epidemiology models of infectious disease I was showing before, that kind of have like, yeah, this is kind of a good reference of biological models. So you can have a look in there if you want more examples of this sort of modelling. So just to briefly mention some terminology, I'm not sure if I'll give the best definition of this, and it feels slightly woolly in my head, but it's some words that I think are useful to know. So on the left, and kind of what we've just talked about, mechanistic models. So here we write down the exact mechanism by which we think a complex system or the biological system behaves. So we're saying this reacts with this at this rate, which then leads to this reaction. And so you're kind of using your chemistry there, or your biochemistry, or in biology. You might talk about predator and prey and how they interact. But you write down sort of all the rules of the system. Something that's very popular that I'm actually not going to talk about today. This kind of would really be the focus of another talk, which I'm sure you'll have that either in this course or would have had it in the introductory, your first year course in Heidelberg. But just to mention non-mechanistic models, which kind of assume something quite statistically general about the system. These tend to be very general, flexible models that fit many data types. So the most well-known of these, I would say, is linear regression, where you assume linearity between an input and an output variable plus some error. You do actually assume a distribution of those errors. But in general, that can fit many types of data, but it doesn't really tell you anything about how or why this response in X creates this response in Y. And then, of course, these can become much more complex. So you probably, I'm sure, are all aware of large language models. So this is an example of one block of a GPT model. So each of those blocks there is kind of a single mathematical operation. So you can see that already looks very big. But I think GPT-2, which was the one before the first ChatGPT, had over 96 of these connected together. So these are huge systems with lots and lots of parameters and are incredibly flexible and can fit lots of types of data, but it doesn't really tell us about how the data is generated. That's kind of the difference. And these models at the bottom tend to be called black box models because we can't really look inside them very well and see how they generate input from output. If you're interested in this kind of thing, a book I'd highly recommend is called Introduction to Statistical Learning. That covers these kind of traditional non-mechanistic models like these squared regression, and does also go all the way to deep learning. And they sort of, the way they define, yeah, you can see my cursor here. So this is the linear regression model. And then they sort of define this flexibility versus interpretability about how black box things are. So as you go down this way, things get more black box. And you can see deep learning is very flexible, but not interpretable. So that's a great book if you're interested in that. But today we're kind of going to focus on mechanistic models, which they do have some overlap with non-mechanistic models, but the sort of thinking of how you design them is quite different. So how do we design one of these? So here's our first example of a biological model. So this is going to be an individual-based model of alleles or in a population. So this is a Wright-Fisher model specifically, and here I have 4 different alleles, A, B, C, and D. And I'm going to have a constant population with non-overlapping generations. So something that is actually kind of reasonable for would be something like birds who lay eggs every year. And a number of those offspring survive into the next generation. So if I have A, B, C, and D, and they're all equally fit, and I have 4 individuals in the next generation, what I want to do is I just want to randomly pick from the next, for the next individual, 2 parents, a mother and a father. I'm actually going to keep this simpler and talk about bacteria, which is also what I study. So actually we don't need 2 parents. We just have one that's going to clone itself, and some of its child cells will survive. So if I run that for one generation, it looks like that. Of course, there was some random draws there, but in this case, D has died out. None of its offspring survived to the next generation, but B created 2 offspring. And I can keep doing that forwards in time. And I get some randomness there. And what I plotted on the left is the frequency of one of those alleles. And you can see this gives me genetic drift. So actually this model is a description of genetic drift. So these individual based models, in a way, I think they can be the simplest models to formulate. And I think often how we as biologists tend to think of things, we tend to think of individuals and how an individual behaves. That might be an individual cell, it might be an individual of a population, kind of depends on the system. But you can think about how those individuals change in time. Again, I guess I'll just quickly mention that mostly we're talking about time-based models today rather than spatial models. So you can make this system more complex. Another thing we can do very easily in this sort of computational framework is we can add in selection. Fitness, usually called S in population genetics, where an individual has an increased or a decreased chance to be successful. So you just change the amount of offspring on average they have in the next generation. And here we've got an example with positive and negative selection. You can see the stronger. So on the top, I've got positive selection and I make it stronger. This allele quickly comes to dominate, but in the left, drift can still happen. So actually it still sometimes gets purged. And then in the bottom, you can see where it's got a negative, so it's less fit and it tends to die off. This is something that would actually be quite difficult to solve analytically, but with doing it computationally, it's actually relatively easy because you just change the probabilities. You can also do that in kind of arbitrarily complex ways. So you can make those probabilities dependent on time. You can make them dependent on frequency of different alleles in the populations. And you can really, with these individual based models, you can make them incredibly flexible and introduce all the different mechanisms you want. The issue with doing this is that the computation and the statistics gets quite hard. So computation as in how long it takes to run these models and sort of doing it correctly. The statistics is the fitting to data. So we'll just talk first about the computation and we'll come on to the statistics later on. So the kind of common way you might try and simplify the computation here is you might look at the states of the system. And you actually notice that it's fairly simple. There are only 4 states in this system. You can either be A, B, C or D. And there's nothing on top of that. So if I had 500 individuals, rather than keeping track of the 500 and all of their individual states, what I could do is I could just count how many of those 500 are A, how many are B, how many are C, how many are D. So here in this simple example of 4, it doesn't look like you gain much, but so I've done the sums here. Yeah, so yeah. At first there's 1 of each, and then here I've got two A's. And 2 Bs, but that really, if imagine having 100 million individuals, which if you're keeping track of cells would be realistic. If the cells only have 10 states, just counting the number in each state is going to be much more efficient than simulating 10 million forwards into the next generation. And that kind of leads us on to compartmental or state space models. So I'll walk through an example of one of these, but these kind of lie on this spectrum. So we've talked about individual based models here. So this is our Wright-Fisher model, which is very flexible, but it can be quite difficult to simulate. And here what I do is I keep track of every individual, and for each individual I keep track of their state, and their state can be very complex. So here I might have their age. So states are often kept track of by these bit vectors. So here I'm sort of saying whether they're a child or an adult, for example, and then this might be whether they're vaccinated or not. And I need to keep track of a lot of information and simulate all of that forwards. On the left here I have deterministic compartmental models. So that's kind of simplifying the state or just summing the states together. So here I've got an example of an infection where people can be susceptible, say to COVID, being exposed. Once they're exposed, they become infected, and then they can recover. Then after some time, they become susceptible again, and that's a reasonable and simplified description of that system. One thing I will mention today is we can sort of get somewhere in between these two regimes by making our compartmental models stochastic, and I'll mention how to do that again, just kind of as a way of introduction. So let's see if I can play this video. Yes, so this is an example of an SIR model run in space. So you can see here that I have, I think it's 100 individuals that move around randomly in this box. They start off as blue, which is susceptible. There's a few infecteds who are red. And then after a certain amount of time, these red individuals recover and become grey, so they can't infect people anymore. And so in this model, any time that a susceptible, so a blue person, and an infected, a red person come together, there's a chance, which is kind of the secondary attack rate, that they become infected. And so if we just run that again, so we start with a few red points, and you can see that as they bounce around, the blue points that they hit, a few of them also become red. So the number of reds grows, but then over time the reds recover, and you see this kind of typical epidemic trajectory curve, which we'll see drawn a bit more clearly in a next plot. So you can imagine how you would simulate that. You're kind of moving all the positions around, and at every time point you see which points are close together, and you might infect them. So that's actually a spatial model where we're keeping track of space there, and it's also an individual model. We're keeping track of every individual. Though we want to make that simpler. So how can I express this system mathematically? So it would be difficult to write down as a function of time and also infection rate what the number of susceptibles look like, or the number of infected. That's going to be our kind of typical epidemic curve that looks like this. So might have, this is infections as a function of time we saw before. It looks a bit like that, but that's not like any mathematical function we know, and it's not really kind of possible just to write that down. What we often can write down, and this is true of these kind of systems and also biochemical reactions, is we can write down rates. So here is kind of the fundamental part of an SIR model, which is I'm writing down the rate at which susceptibles change. So the rate at which susceptibles change is the number of infections that occur per unit time. So that's the ds by dt. So change in susceptibles over a change in time. And obviously that decreases, so that gives me this the minus sign here. And then I have the infection rate here. So this is the chance when a susceptible and an infected come into contact that infected person causes an infection in the susceptible. And then here is the kind of key bit. So it's the number of susceptibles multiplied by the number of infecteds. And that kind of makes sense. So the more susceptibles I have, the more people there are to infect, so the more of these interactions are happening, and the more infected people there are, again, the more chances for infection. If you actually want to sort of derive this more carefully, it comes from the law of mass action, which is that you have these particles moving around, so particles in an equation sort of treated the same way, and that they come into contact at equal rates, and then you have this normalizing factor for the size of the population, which is just needed to keep this ratio here correct. So that gives us the full system of differential equations. So you might remember from maths courses you've done in the past your ordinary differential equations, and this is indeed an ODE model. So this is the equation we had before. Now we need to write down equations for the other two states. So here are the infecteds. So this is just going into this partition. So this is just added in here, so it becomes a positive term. And then the one other thing we do is that we have a recovery rate. So each infected person, at any given unit of time, they recover with a rate gamma. So these go into here. So this is the I to R transition. So really I kind of have two different changes here. And then if I add all of these together, so I've got the total size of the population, N, is S plus I plus R. So dN by dt is the sum of all of these equations here. And you'll see that this sums to 0. So the population size doesn't change over time, which is correct. So what can we actually do with these? So you might, I'm sure you have done ODE systems before. You can try and solve them analytically. So you can try and integrate them essentially. So remove this dt. So you get the functions of S, I, and R over time. It actually turns out for this system, even though it's simple and it's linear, there is no analytic form for these expressions in general. One thing you can do, which is easier, is you can analyze the stability. So if you set all of these equal to 0, so that's where the system doesn't change, so that's kind of the steady state of the system, you can solve these equations algebraically. You can find the steady state. So in this system, it's where there are no infections, so at the start and the end of the epidemic. And then you can also analyze the stability of those points, so whether they remain stable or are inflection points. But often for biology we actually do want to get the solutions over time, and we can solve those numerically, so that's by doing some form of numerical integration. So in R, there's the differential equation solver package. In Python you've got the ODE integration. In principle, these use something quite similar to the trapezium rule, with a few kind of clever tricks added on to make them more general. But you can look at the rates over small times and see how they change, and get computational solutions to these equations. So this is known as an ODE model, an ordinary differential equation model. There's partial differential equation models, which I won't go into in more detail here. So if we actually do this for this system, this is an example provided to me by Leone, who you'll hear from later, of integrating these over time, and you get this nice epidemic curve here. So this is the number of infecteds, susceptibles decline here, and then the recovered is the other line. And you can see this is quite similar in shape to our stochastic simulation from before. So that's all great. You'll notice this is very smooth. So one thing this is assuming is that this is a continuous variable. So it's not taking into account that the population is discrete and individuals move in actually numbers of one of counts. And it's also deterministic. So every time I run this forwards, I get the same answer to my differential equations. And that is a property of ODE models. So life is, of course, stochastic. We know that the chance of infection is probabilistic. And so one thing we might want to do to extend this is to have the same assumptions of the model, but allow some, you could think of it as noise, but just kind of randomness in the infection process. And the standard way of doing this is to formulate it as a continuous time Markov chain, which in continuous time Markov chains, you have you end up with these kind of terms where you've got 1 e to the minus probability of transition times time. And this is familiar from before. Then I have that probability over a number of people, so I end up with binomial draws. So for each susceptible, I move them to infected with this probability. And then that, if I run that forward, that's the CTMC model. If I run that forwards in time, you'll see similar shape, but much more variance in the trajectories, which has a few advantages and often fits data better because data is noisy as well. So I'll just briefly mention that there's different ways to do this. I think I'll just skip over this quite quickly. But one example of how you might use these models or write these models in practice is to use a package. And one that I worked on a bit in the past, but really developed by Rich Fitzjohn at Imperial in London, is the Odin package, and this lets you write models kind of in the way we've just described rather than in code, and run them quickly and fit data to them efficiently. So here's an example of what the code looks like. You can see it's basically what I put on the slides before, so it does look like code but doesn't have some of the other syntax. And we define some of the things we said before. We've got the total population size, we've got our chance of infection, we also have the initial state, so the size of the population, and then we also have the parameters, so the rate of infection, the rate of recovery, and then the initial sizes. So that's all quite readable. And then in R code, so this is actual R code, you would just load this model in here, and then you can give it some initial values. You tell it how much to simulate on how many threads, and then you just run it forwards, and you can get that plot from before. So you might wonder, why do that? It seems pretty easy to write this out, but this becomes really important for big models. So the model I showed you before of COVID, which is called SIR-COVID because it's an SOR model, but here I've got the sort of S partition, E partition, and then things get more complicated. This is the I partition, and then this is hospital and recovered. So the number of partitions grows, but it is basically an SEIR model. And in this I have 10,000 compartments, about 100,000 transitions, because I've got lots of different age groups, I've got vaccination status, I've got PCR tests. Then it becomes really helpful to write this out in one of these packages, because what it's doing is it transpiles this into C++ code, which firstly can be run more quickly, but also it kind of keeps track of a lot of the tedious bits of writing these loops out. So you can see these really become quite complicated, and getting all of these indices right is hard. So if you do develop these kind of models, if they get more than having a few ODEs, you probably want to consider using a package rather than writing out the ODEs yourself and using an ODE solver. And yeah, actually, this is an early version of the model, but it ended up as 18,000 lines of code, generated from more like 500 lines of model code. So you do save a lot of time by doing it this way. So that kind of is a bit of an overview of a really introduction to, rather than an overview of, how you might formulate models. I'm going to spend the last bit talking about how you fit those models to data. So what I've told you about so far is, once you've got this model, how do you simulate it forwards in time and look at how the system evolves. But often you actually want to infer things from your data. So you might have done some experiments, have some observations, have some sequencing, and you want to know, first of all, is this model a good description of this system? And then if it is, what are the values of the parameters in that model that explain the data well? So sticking with the example of the SIR model, we talked about the beta and the gamma, the infection rate and the recovery rate. But of course, in a real outbreak, that's not something we observe. It's not even something that's particularly kind of a real biological thing in a sense. It's not fundamental, but it is useful. So that's kind of again going back to the initial broad description of modelling, something we want to use and is useful to know. But there's no experiment we can do to measure the infection rate. The recovery rate we probably can measure, but again, it's difficult. What we do observe in an epidemic is the number of new cases each day. So especially for smaller epidemics, we can keep track of every single infection. And this is actually simulated from the model, but even in simulated data, it looks quite noisy. So you can see clearly when the epidemic is underway here, you do get lots of cases and then they tail off. But what you can use model fitting to do in this case is you can say, if I have this epidemic model, how likely am I to create a case count distribution that looks like this, given some beta and gamma? And then with that, I can say which beta and gamma makes it most likely that my case count distribution should look like this. And that's kind of the essence of model fitting. The way it's done in practice is usually through Bayes' rule. So you've almost certainly come across this before, and I think a lot of focus is given to the prior, and that is a fundamental difference between Bayesian and frequentist statistics of saying for some parameter, even before I observe any data, I'm going to say it has a certain probability of existing. But actually what we're going to talk about, the prior, is not necessarily that important, and you can basically ignore it. So you can use things called improper priors, which do actually completely ignore the prior. The important part here is the translation between the posterior and the likelihood. So what I want to know when I fit a model is what's the probability of some parameter given the data. So if I talk about the infection rate, what's the probability? Well, let's use R. So what's the probability of R being 1 given this observation of a time series? That's obviously a very practical question. Now, I can't get that directly from my model, but what I can do is I can write down a likelihood. So what's the probability of observing this data given some parameter? So basically I can make a guess for the parameter and then say how likely is it that I get this time series of data. And I can use some sampling, which I'll mention in a minute how that works, to translate between sort of this useful thing that I want to know and then this thing which I actually get in the model. So how might you write down a likelihood? These can become quite complicated, but in essence they're normally built up from simple parts. So if you see likelihoods written down in research papers, they'll often be these big complex algebraic expressions, but if you break them down into their parts, they're often quite simple. So I put on the left here how this is. So this is actually in the Odin code from before. So more there for reference. Sorry for that atrocious use of the pointer. But anyway, there's this is, if you want to actually try running this in practice, the code is there. But this is the important bit. This is the likelihood here. So remember that the likelihood is the probability of the data given some parameters. So what I do is I make a guess of some parameters. So I might simulate the model forwards with a beta of 0.2, a gamma of 0.1. And then here, this is the case series I get out of the model. So I run my SIR model, and then here I look at the change in I. So this is I from the day before, and I in the new day. That gives me the number of new cases. And then what I can do is I can look at, in my actual data series, how many cases were actually observed. So this being this way around, the expected number of cases from the data is the observed number of cases. So this gives me a lambda in a Poisson distribution. So then I say, what is the chance of actually getting this number of cases seen in the model? So if I have counts, basically I want to use a Poisson. If they're over dispersed, I may use a negative binomial. If I also wanted to have, say, deaths in the model, so some number of infected people die rather than recovering, I could have a separate Poisson likelihood for the counts of deaths, and then I would just multiply these together because probabilities multiply. And often what you'll see is the log used, because when you make these from pieces, it's easier to add the logs, and it leads to fewer problems with computer precision. But basically, I simulate the model with some parameters, and then I look at how well this fits the actual data series observed. And the kind of trick in modern samplers is to get rid of this term. So this denominator here is difficult. It's called the evidence. It's quite pesky to evaluate, because what you need to do is you need to do an integral over all of the possible parameters in your model. So when you have, even with a single parameter, it can be difficult to do that integral numerically, but particularly when you have many parameters, looking at every possible combination of parameters and evaluating all of their likelihoods becomes impossible. So what we want is to kind of just make this proportional here, and there's various different ways of doing this. Some are explicitly Bayesian, others are kind of more frequentist, but all of them, I'll just try and highlight this on the slide. I should have put a box around it, but it's kind of the important bit to remember about model fitting. You make a guess of a new set of parameters, so that is some jump distribution often, but you have some way of guessing what the set of parameters is going to be, and then you run your model with that set of parameters and then look at how well that fits to the data. So I could just look at these series and look at the least squares fit. I mentioned that we would use a Poisson distribution, which actually gives us a likelihood. The difference between those two things is approximate Bayesian computation just looks at distance from the data. I don't have time to describe that in general, but that is quite a nice approach to fitting biological models, because you don't need to evaluate a likelihood. You just need to say, does this look like my observation? So that's becoming quite popular, because you can make your simulation of the data as complex as possible. As complex as necessary. You can sort of give a full biological description. But sort of thinking about likelihood methods, if you can write a likelihood down, you can use maximum likelihood. So here in the posterior distribution, I would find perhaps this value here. So this is the probability, and then this is beta in the model. Maximum likelihood is going to make guesses for beta, which just follow. So this is called hill climbing. They just try and move up to higher probabilities and then stop when they reach the maximum. So that's the mode of the posterior distribution. And then we get this value for beta. So this is our beta estimate here. A slightly better way statistically of doing this is MCMC. So this samples this posterior. So it doesn't just try and find the highest point, it also tries to find the whole shape, so you get this whole distribution. And then this is useful because I can also take the 95% credible interval here, so I can get an error estimate on the parameters. And then I'll just mention quickly something that's become very popular, which is Hamiltonian Monte Carlo. So this is an optimized form of MCMC. What's made this difficult in the past is, as well as the likelihood, you need its gradient, which can be very tedious to evaluate. But there's now, basically thanks to deep learning, ways of automatically finding gradients computationally. So just to run through quickly, again, kind of don't have a huge amount of time to go through this, and it's a real topic on its own, but what if you ran MCMC, it might look like. So here, if I have two parameters, I would start with my guess over here, and then I randomly move it around. When it's a better likelihood, I take it. If it's not a better likelihood, I still take it with the probability given by the change in those two likelihoods. And that's called Metropolis Hastings, Markov chain, Monte Carlo. Often people just call this method MCMC. It's very simple to implement. You can statistically prove that that gives you the correct estimate of the posterior distribution. But what you find is at the start, there's this kind of trajectory of your samples. This is known as the burn-in as you get to kind of the correct parameter estimate. So here the correct parameter estimate is 0, 0 for X and Y. Once you get there, you then sample around it and find the right distribution. And if I plot kind of, this is looking at this side on, and this is looking at this side on. So these are known as the marginal posteriors. So this is for a single sample. So you can see there's this burn-in period. and then you get this sampling. And when this sampling has gone well, this looks like a fuzzy caterpillar. You'll hear people say, and it does look like a fuzzy caterpillar if it's gone well. So again, you probably, rather than wanting to use this yourself, you probably want to use a package to do it. So MC State is the one that plugs into Odin and Dust if you're using those packages. But some good ones in are, you can use the Coda package, and then there's PyMC3 in Python that let you do this. If you want to play around with this, there's an example listed here, but there's others online too. So if I run this MCMC, so this is with a true value of beta of 0.2 and gamma of 0.1 in our SIR model. One thing I didn't mention is that in the simple SIR model, the R0 is just beta over gamma. So the number of infections each person causes is the infection rate divided by the recovery rate. So with these parameters, this is an R0 of 2. So that correct value would be here in the distribution. And you can see I've run a few independent samples here, but you can see they bounce around the correct values here of 0.2 and 0.1. And then that gives me my posterior distribution for R0. So I've used an infection series, an incidence series, which is actual data, and my model of how the population evolves. And then I get my parameter estimate here. And also you can see I'm getting some confidence interval, and it's actually non-symmetric, so I can say how confident I am in this estimate. Plotting that in kind of terms of what we saw before, so if I run it forwards with the correct parameters, you can see that the actual SI and R time series in the model, they sort of fit pretty well to what I got in my stochastic trajectories. If I pick the wrong values, so that's this of an R0 of 4, you can see I go far off, and particularly the number of recovered is far below what would be expected, and I see correspondingly this one is less likely here. So I move away from these parameters and move to these, which are the correct ones. Unfortunately, one of the big difficulties of this form of MCMC is that you can see that is kind of called quite sticky. Each of those samples tends to stay in one place and is quite blocky. And I need to do some tuning of what the guess distribution is. So I haven't talked about the guess, how you make the next guess for your set of parameters. But that is something with kind of standard MC you need to play around with, and it's quite frustrating to do and get right. So this actually gives a much better estimate of R0, and these are much better chains, but it required some kind of changing to this how you pick the next guesses. So if you don't want to do that, you want to use Hamiltonian Monte Carlo. Now again, I don't have time to go into this in detail, but just wanted to put it at the front of your minds. If you can use STAN, I think is a good rule of thumb. So STAN is a probabilistic modelling language. You can write down all sorts of models in there. It will do MCMC for you, infer parameters from data without you needing to do any tuning. It will also tell you how good your model is as a description of data. So this is a totally different example, but if I had some data like this that was kind of quite lumpy and had different peaks, and I think there are different generating processes, so I think there's 3 Gaussians centered here, here and here with variances, and my points come from one of those. And this is quite common in lots of biology. So an example would be sequencing data. You might have this being error k-mers, this being real k-mers, and you want to tell those things apart. With STAN, I can say, okay, well, I'll put 3 Gaussians with 3 different means on there and move them around for me until they fit the data quite well. And what STAN will give me is it'll give me that sampling, so that's a nice well-mixed chain. And on each of these I get not just where they're most likely to fit, but also some error. So STAN is great. It's quite easy to write down the models. It's kind of uses one of these domain-specific languages like Odin did, so you don't have to implement the sampler. You don't actually have to kind of get into the statistics much yourself. You just write down the model. So another thing STAN is very good for, in I think kind of a work a lot of us will be doing, is doing sort of funky regressions, so regressions with unusual output data. So you might have come across Poisson regression before. So this is where your output variable is a count. So rather than it being continuous, and this is important at the bottom here because I can only have 0, 1 or 2. In this case, I'm looking at the number of laws passed via versus different kind of political and affiliations and demography in the US. But if you've got count data, you probably want to use one of these forms of regression. So the standard regression here, it sort of goes wrong and gives you predictions of minus laws, whereas the Poisson data, well, first of all, it fits the data better, but it gives us sensible predictions. So there are packages in R, for example, that let you do Poisson regression, but it's much easier to do these kind of changes in a language like Stan. And there's lots of documentation online of how to do that. So just to sum up, I've given you a very, hopefully given you an overview of what modelling is possible and maybe how you do modelling. It's a huge field. There's lots of things. This is kind of, even just in SIR models, things we could do that we haven't talked about. So adding extra partitions, we mentioned briefly, we might want to model hospitals better. Adding delays, so reporting delays. For example, a PCR test might take a week to come through, so how we account for that. We have looked at time series data, but I haven't talked about the complexities of modelling long time series. Spatial models we very briefly touched upon, but have basically kind of ignored. I did mention mixture models, but really in no detail. Hidden Markov models are historically very popular for sequence data. I guess transformers might be becoming more popular now, but hidden Markov models I think will still remain useful. Phylogenetic models and discrete parameter spaces, so where your parameters can take only a set of values, we haven't talked about, and many more things. But anyway, I hope this has been a useful jumping off point if you haven't kind of done this sort of modelling before. So just again in the summary, if you were to want to develop your own mechanistic model of a biological process, how might you do this? So first of all, decide what do you need your model for. Am I going to try and fit some parameters? Do I need to use data to fit parameters in the model? Am I going to use it to describe how a system might behave? Do I want it to make predictions or forecasts? Or am I going to do counterfactual? So if I change the parameter, how does the system behave? Those are the kind of things you can do with models. But think about that before you start the modelling work. If you can, start with an existing model, even if it doesn't exactly describe your system. So if you're looking at a population, probably start with a Wright-Fisher model. If you're looking at ecology, probably start with a Lotka-Volterra model. If you're looking at epidemiology, probably start with an SIR model, and then make it more complex from there. And then add that complexity bit by bit. So start adding compartments. So rather than trying to do it all at once, like many things in science, but split the problem into smaller and more doable pieces. Run your models forward in time, plot the output, look at it. That's always useful. See how it compares to the data. Is it getting it right? And then sometimes people rush into fitting, but only try and fit the data once you're happy that the model is a good description from the system. And actually, even before trying to fit to real measurements that you might have taken or real biological data, just simulate from your model. So simulate data from the model and see if you can fit the model to that, because that's the kind of best case test. And just be aware that fitting to real data is always hard and will usually require multiple rounds of iteration of the model. So I'm not going to mention them. I think I've mentioned one or two throughout, but you'll get the slides. So if you want to look into any of this in more detail, here's some resources, including the machine learning textbook I like, which I haven't talked about today. Here's also some pets, Maggie and Martha. And finally, here's the group. I think you'll hear from Leonie later, who's been doing some nice work on modelling. Yeah, thanks to everyone. It's great to have people work on these projects. Just a reminder that if you want to talk about modelling or ask me some questions, I'm very happy to answer by email or arrange a session later. So I think that's everything from me. Yeah, sorry I'm not there right now, but I hope you enjoyed the rest of the course and I hope you found this useful.